So my official title is Transforming Medicine Through Engineering. But what I'm really going to talk to you about are some lessons learned in skeletal muscle design and plasticity. But if I was really being honest with you, my title would be I Love Sarcomers. And that's because since I was a scientific boy, I've been fascinated by how muscle looks and how it works and tools used to study it. So for example, if I show you this picture, maybe we can put the lights down a little bit. You can see that skeletal muscle is this amazing composite tissue. And let's see, do I have a pointer? I do have a pointer. Sorry. Sorry, muscle is this great composite tissue. And what I'm showing you here is a cartoon of muscle at the micro level, which is the front of the screen coming out to you. And here you can see the well-known contractile proteins of muscle that are myosin and actin. These are the myofilaments that power force generations that allow you to move. And it's really amazing that these molecules generate piconewtons of force and move nanometers, and yet as humans, we can generate giant forces and have giant excursions because of this incredible arrangement of these molecules. And then way in the background, you see muscle sort of more as meat, which is the stuff you eat, and is composed of muscle cells embedded in connective tissue. And I would say more and more, these days, we've become interested in connective tissue. And so I'll just show you a pretty picture that Alison Gillies, who's a Ph.D. student, recently generated of a fiber. And we think these fibers have really interesting connective tissue patterns on top of them. And here you can see a little bit of it. But you can see if you were an engineer trying to understand a composite tissue, this would be a great one to study because you've got little motors embedded in force-generating fiber composites that then end up generating force in the outside world. And I'll tell you, in terms of muscle diseases, the muscular dystrophies, etc., these are all diseases of force transmission. Hardly any muscle diseases are diseases of force generation, the contractile proteins. Most of the problems in muscle happen when force is transmitted through the cell and outside to the outside world. Now, if you take a slice down the middle of one of these cells, you see a pattern that looks like this. This is what we show in most of our textbooks. Muscle is a crystal, really, and these thicker bands, the so-called A bands, contain the contractile proteins, and these thinner bands, the so-called I bands, contain force-transmitting proteins, and actin and myosin are what do power muscle contraction. So people have known about muscle having the striated pattern, and that's why it's referred to often as striated muscle, for a long time. But the trick is, until maybe the 40s, 50s, and 60s, the significance of that striation pattern wasn't well understood. So I'll talk about that a little bit today and some of the clinical application. I'll also tell you that if you're an engineer and you see this banding pattern, you can start to drool a little bit because periodicity in engineering can be quantified. And actually my advisor was one of the first guys to take a laser beam, which is obviously now like my laser pointer, but in the good old days it was less common, and shine lasers into muscles. And because most of the visible light lasers have wavelengths in the micron range or submicron range, and since these banding patterns are about 1 or 2 microns in diameter, you get an interference pattern between the incident laser light and the proteins. The cool thing about that is that interference pattern lets you measure microscopic structure in the muscle with a laser. And so one of my earliest exposures to some of this stuff was developing optical theories that described how the intensity of light changes as you go through a muscle. And of course you come up with a big damped sine wave that is a mathematical representation of this picture. And so you can see here this damped intensity profile as you scan across the screen. This would be a picture taken. If you just put a muscle, say right here on the screen, and shine a laser straight up, you get this interference pattern. And of course, because the muscle has a periodic structure in it, the peaks of this pattern occur at regular spacings that are described by the grading equation. And so we spent a lot of time studying muscle optical properties to try to understand what three-dimensional information we could get from muscles. And we used a tool, model system to study this, and it was the frog muscle. I say frogs are God's gift to muscle mechanics because you can isolate single intact frog cells and they work just like muscles. Whereas you try to do that in some of these mammalian tissues and it's almost impossible because of the incredible connective tissue. So in the 60s and early 70s, the single frog fiber was the muscle studied and this banding pattern, now you can see it here, the banding pattern at low magnification, turned out to have significant impact on understanding the mechanism of muscle contraction. And that work, which was done by Andrew Huxley, who already had a Nobel Prize and was going for Nobel Prize #2, which is shown in this relationship. This is the length of the individual contractile units of muscle, the so-called sarcomeres. Sarco is the Latin prefix for muscle. And the vertical axis is force. And what you see here, the solid line, is the relationship between force generated by a muscle and the length of its sarcomeres. So when muscles are stretched a lot, they don't generate a huge amount of force because these filaments can't interact. And if muscles are shortened a lot, they don't generate much force because those filaments kind of interfere with each other, but there's an optimal length. And so my PhD thesis had to do with this topic. And I remember I went to Mecca, which was, at that time was Cambridge, and gave this talk to Andrew Huxley and was ready to hear what a genius I was and how important these insights were that I was showing them. And he said to me that really the entire body of work that I had generated was sort of kind of an experimental artifact. Because he says, we know that muscles are designed to operate on the plateau of this length tension curve. So things that happen here and here are really sort of an academic. And I kind of was depressed and disappointed because he didn't call me a genius. I came back and I started thinking about this. And I actually got my PhD in a zoology department where people were studying fish swimming and frog hopping and bird flying and anteater digging and real functional stuff. And I was starting to ask guys, well, where do muscles operate in vivo? And put graphically, it's shown here. Here's the length tension curve. Here's how muscles operate. But if you're designing muscles as an engineer, what is the best place for a muscle to operate? And why would you pick one place over the other? So the more I studied it, the more I realized we didn't know the answer. It's like, I should have known that because when he asked me, I should have been ready with my answer. And I wasn't. And a buddy of mine was at SeaWorld, Reggie Edgerton, and he saw these rainbow-looking little tropical fish, the so-called glass catfish. And the interesting thing about these catfish is they have this rainbow pattern. They have transparent skin, but the rainbow, he just called and said, now how do these fish get a rainbow pattern on their skin, and why does the rainbow pattern move? And of course, I'm a diffraction freak at this point, and I go, it's got to be white light diffraction. So I got in the car, I took my laser, I took a flashlight, I drove down to SeaWorld, and I went to the front of the SeaWorld, where they charge you $1,000 to get in. I said, I gotta see, the glass catfish. And the lady just looks at me like, okay, that'll be $200. No, I'm a scientist. I need to study your glass catfish. She called some people. Finally, I got in there and we started looking at this glass catfish. And I literally had a giant laser at the time because it was the old days. And I said, can I just shine this laser through? I just want to see if I get a diffraction pattern from your fish. And she knows I'm crazy. She goes, all right. So I plug in the laser and I start to turn toward the fish. She goes, well, don't kill it. I go, no, it's just a little light like this. It's not going to hurt the fish. I shined it through. I got this great diffraction pattern. And then I immediately went to a fish store, a pet store, and bought a tropical fish. I bought one, and this is him. And I trained him to swim in this little, what we call a fish mill. Instead of a treadmill, and he learned. It was tricky to train him, and he had to train us a little bit, but we used one fish for our entire lifetime study of these fish. And you can see he's sort of in front of this laser, and he's swimming around, and he had to be the right speed. This bubble turned out to be critical. And the fish anatomy is really complicated, so it turned out that when you actually look at the fish, it looks like this. But when you look at his diffraction patterns, the fish looks like this. And so this is him swimming. You can maybe make out a little bit of a vibration in there, and you see how the angles change every so often because it's moving around to different populations of fibers. So I was big into data acquisition. I had made a deal with Intel to get their high-speed A to D converters, and we made a two-dimensional array that would transduce the angle and spacing and all this kind of stuff. And ultimately, it led to, after I'd say we probably took 50 hours worth of data and probably got 15 seconds worth of usable serial data because we could bin things over time. And we showed that these fish sarcomere length changes were very regular. And again, if you're a sarcomerologist like I was and am, you get really excited by these numbers because these are really tiny numbers. And if you can see this change in length, 1.87 microns to maybe 1.94 microns. If you put that on a length tension curve, you see that fish are designed to basically vibrate back and forth at a little teeny sarcomere length change. So you have muscle on one side and muscle on the other side of the spine, and they just wiggle back and forth just a teeny bit. And this was the first demonstration that first of all, of sarcomere length in a moving animal, but also that there would be some kind of physiological significance of the portion of the length tension curve that was utilized by an animal. And fish doing, since these little catfish do basically that same thing all day, they had a very narrow range, they were highly specialized. Meanwhile, I have a buddy who's here on sabbatical here. He was a hand surgeon, but he wanted some muscle training. He and I met each other, a guy named Jan Frieden. He was turning into a pretty serious hand surgeon, and we were really interested in doing this in humans, because in humans, if you have a muscle that's very length sensitive, first of all, you'd like to know where it's operating, and second of all, if you're doing surgery and you're changing the length of the muscle, you could easily alter the function. So he and I developed a tool that we could take into the operating room, which was basically a fancy laser. A laser beam with a prism that we could stick into a muscle, a photodetector that had a photodiode array on it, and an interface to a computer. And we wanted to go ahead and start measuring sarcomere length in people. So again, we were pretty young at the time trying to talk people into it. So the deal we made was we would take this laser to Sweden, which is where he was working in northern Sweden, and he would do laser diffraction on me, and I would do laser diffraction on him, and we would get together, and we would, we were studying wrist muscles at the time. We would get together, and we would show that, we could measure these lengths, that they would be reasonable, and that they would provide useful information, and that we wouldn't die. So that was the deal. We had mathematically modeled this muscle for years. I had dissected maybe 50 ECRBs, extensor carpi radialis brevis muscles, because these are the muscles that get tennis elbow. And then you can imagine my joy when I saw my own personal ECRB. And we started moving the muscle, and we started seeing things that we hadn't predicted based on our models. I mean, we were testing the model because, here's flexing and extending the arm. There's the muscle moving back and forth. And we saw a little peak of a tendon out here in the corner. We had done tendon mechanics too, and we thought, you know what? We just don't have enough to see. We don't have enough exposure. And so we expanded our system a little bit. And here now you can see the tendon. This is the only gross stuff I have. A tendon and the ECRB muscle. Sorry, I should have given you some warning about that. And now, if you start flexing and extending, you can see huge amounts of movement, and more importantly, huge amounts of fiber rotation, which were not part of any biomechanical model, had never been transduced before, and it really revolutionized our thinking about how these muscles move in vivo. And so we did a bunch of experiments on normal patients and started looking at surgical patients, and basically developed a small version of this device that we could use. This is the last gross paper. Developed a surgical device that we could use and do use during hand surgery. It's reconstructive surgery, and you can see here's a bundle of muscle that's being isolated. This is a real life patient who's having a muscle move from one place in their arm to another place. They've had a spinal cord injury, and therefore they can't, for example, grip their hand, but we're taking a muscle that works near their elbow and moving it with an extension cord that we take out of their legs, out of tendon. And here you can see the muscle is being diffracted, and there you can see the little diffraction pattern, and here's Jan using a caliper to just basically measure it. And you can literally, with a laptop computer and a gizmo like this, you can set their sarcomere length to exactly where you want it to be on the length tension curve. And it pretty much doubles the effectiveness of the surgery. So we've done clinical trials where we're showing this stuff, and I just thought I'd show you an example. Here's a guy who has a neck injury. He's had surgery on one side. On one side, he's had a tendon transfer. He has a little bit of deltoid and triceps function. He could sometimes extend his arm a little bit. It was a little weak there. He has a little bit of extension strength in one of his arms. On this side, on this side, well, that's not him, but this is a guy who's had that same surgery done on both sides of his body. He had no elbow extension function before the surgery, and now he can independently run a wheelchair. And it's only because he's at the right sarcomere length. It's kind of silly. It's such low-hanging fruit. This is the guy I was going to show you. That's elbow extension. And then these muscles, this is taking a little slip of a shoulder muscle and moving it down to the elbow. These are called tendon transfer procedures, and they're extremely successful. They change a person's life because now he can reach out, you can give him grip function, he can feed himself, he can go to the bathroom by himself, he can get dressed, and if he trains himself very well, he can actually get in and out of bed. So you go from, basically as a quadriplegic, you go from 100% dependence on a nurse and aides to independence. It's truly life-changing. So let me skip that. I just thought I'd show you another story. This is a girl who's up in Sacramento working with a former student of mine, Jenny Boakes. Jenny was a medical student here at UCSD and then went on to become an orthopedic surgeon, but we always kept in touch. She did some of the original frog diffraction stuff. So the problem that this girl had is she broke her femur, and she unfortunately broke it in the part of the bone that grows. So once it breaks, that part of the bone stops growing, and you can see here she has a 5 or 6 centimeter leg length discrepancy, which is bad. It's unsightly because she walks with a big limp. It's going to wear out this hip because she's pounding her hip for however many years. Here she's only like 15 years old. And so the surgery that can be done for this is to literally break the bone and put a big frame on her thigh and every day stretch her leg a little bit. That's the good news. The bad news is not everything stretches at the same rate, not everything can stretch at the same rate. So we hypothesized at the time that there was a certain rate limiting factor, and that was the muscle. So we actually went in during her surgery, measured her sarcomere length, made some, again, laptop calculations that are pretty simple, predicted a rate of distraction, it's called, and then after a year, we measured a bunch of stuff, and then after a year, you can see her leg length is pretty much exactly the same. And so again, it's actually pretty low-hanging fruit. The modeling is very simple. The methods are pretty straightforward, but they're the kinds of things that change someone's lives. Okay, I'm going to show you another. This is a young boy. This guy's about 12 years old. He has cerebral palsy. And one of the complications of cerebral palsy are these so-called flexion contractures. They're real bad in the upper extremity. And this was a little boy. Now, by this time, Jan has moved down to southern Sweden. And, he and I have developed a long-term collaboration, but he's working for a lady who is a cerebral palsy surgeon. And, when you have a laser and we're doing all this cool stuff in the operating room and nurses are going, ooh, ah, and students are coming in and we're kind of like rock and roll, in this one OR. She goes, oh, you should come in my operating room and measure these kids with cerebral palsy. And we're thinking, oh, we barely understand what we're doing in these other spinal cord injury patients. Cerebral palsy, CP, is its own trickiness. We said, no, we really don't think we can do that. And so then later in the lounge, she's saying to me, she goes, Rick, I don't think you understand what I'm saying. She goes, I'm in charge of this whole department, and I'm the one who's allowing you to be in these operating rooms and spend an extra half hour doing what you're doing. And on every case, I go, yeah. She goes, well, I really think you should be studying cerebral palsy. And I go, absolutely, I've always wanted to. So we go in, and now we know what we're going to see. We think the tissue's all fibrotic. These things are contractures. When you go in, the treatment, unfortunately, you can see this kid's even dislocated his wrist. The treatment, the muscle is getting shorter or something's happening. He's stuck. And the elbow gets a contracture and the shoulder gets a contracture. So you're sort of stuck like this. The treatment is gross. The treatment is just cut the tendon and the muscle retracts like a rubber band and then you straighten out the wrist, but you haven't fixed anything. So we thought, well, we have these really short muscles. We're going to stick the laser in the arm and we're going to see super short sarcomeres and we're going to tell her, it's like, doctor, it hurts when I do this. And then the doctor says, well, don't do this. We're not going to have any solution. We're just going to show what's going on. So we had made these very good predictions because we had, again, very good engineering models of what goes on with that muscle. This happened to be flexor carpi ulnaris, which is the major wrist flexor. And so we were shocked when it turned out that our prediction was totally wrong. Shocked and excited, I should say. Not shocked because we were wrong. Shocked because of the direction of the change. Okay. Oh, okay, good. So here's normal. When your wrist is flexed, your sarcomeres are kind of short. These are these kids with spasticity. So isn't it bizarre that, first of all, it's great when you have a quantitative prediction because when you're wrong, you're wrong. And that's one of the beefs I have with a lot of biologists. I know a lot of us are biologists too, but sometimes biology is like better, worse, higher, lower. When you have a quantitative prediction and confidence intervals and you're wrong, there's something important happening. So it turned out that these kids had super short muscles, but the sarcomeres inside the muscles were super stretched, which is an unprecedented effect. And it led us down a stream, which we're still working on today, of trying to understand the sort of the biological and mechanical basis of contracture. I can't tell you too much more about it in the interest of time, but I can tell you tools that we've used to try to study it. One is, because we have access to this muscle, we can take biopsies from these patients and we have a piece of a cell. Remember, muscles are multinucleated cells, they're very long. So here's maybe a 1 or 2 centimeter chunk of tissue. You can dissect it down to one cell. This is a piece of a human cell, maybe 2 millimeters long, maybe 50 to 80 microns in diameter. We can do complete biomechanical studies on those. Because we've dissected them carefully, we can still do diffraction on them. So we can do diffraction on these guys, we can do diffraction in the operating room, and we can compare sort of the in vitro to the in vivo condition. And the other good thing about diffraction is if you destroy the muscle fiber, you get no signal. And I like tools that when you've messed up the experiment, the tool gives you no answer rather than some bizarre answer. And over the years, we've been studying muscle fibers and muscle fiber bundles and trying to understand where the changes are. Because if you understand where the changes are, theoretically you can understand where to direct your therapy. And I'll just shortcut a bunch of that by saying, it turns out that not much is going on in the cell, but remember I was talking about the stuff outside the cells. It turns out stuff outside the cells is changing quite a bit. And Lucas Smith, who's a Ph.D. student in the lab, recently performed a study in collaboration with Hank Chambers, who's a local pediatric orthopedist, and Eva and Yvette are both pediatric orthopedists in Stockholm, and a lot of you know Shankar Subramanian from bioengineering, sort of a discovery experiment to do gene expression profiling of these muscles. And the reason this is important is here we have this unprecedented structural change in a muscle. There's nothing you can do to an animal that makes the animal do that. So a lot of times in science and biology, in order to study something, we get an animal model and then we can study it. This is a uniquely human problem. So the nice thing you can do, you can take that human muscle biopsy that we used for mechanics, and you can look at the various families of genes. This is supposed to be a schematic muscle. Here's the outside of the cell, and you know that muscles are associated with nerves up here. There's contractile machinery in here. There's calcium regulatory stuff over here. There's all the extracellular stuff over here. There's all the growth, sort of hypertrophy, atrophy pathways. And you can study these pathways and try to figure out how is the system confused, and then you can go ahead and try to understand what can be done to fix it. So this is the kind of thing that can be done. I would say it's sort of the perfect combination of biology and engineering tools. Use the engineering tools when you need to know things that are quantitative, make predictions, test hypotheses. Use the biological tools, I would say, more for discovery and maybe development. I did want to show you one other sort of area that we're really interested in. Another application area is sports medicine and how muscle gets injured and exercised and strengthened and all that kind of stuff. Now, the good thing about that is there are animal models for studying muscle in exercise. And here in the lower right-hand portion is a picture of a rat. The perineal nerve has an electrode around it, and the perineal nerve, when it's activated, dorsiflexes your ankle. So the ankle is trying to come this way. But we have this guy under anesthesia, and we're forcibly plantar flexing his ankle while he's trying to dorsiflex. And it turns out when you stretch an activated muscle, that's the best way to injure it, and it's the best way to strengthen it. And that's why sometimes people say there's no pain, no gain. And we're trying to understand what's the molecular and structural basis of no pain, no gain. Is it really true? Do you really have to injure it to make it stronger? So we're studying that, and there have been a number of studies in this area. And I thought I would just show you one that has promise in a bunch of areas, and that's studying this one particular protein. And I'm telling it because I know a lot of what we're supposed to do here today is talk about career guidance, and I want to tell you about an accident that I had happen. Not a bad accident, just a mistake I made. I was in Sweden, up in northern Sweden again, where they had a great muscle anatomy lab, and the fridge was full of antibodies. And so one of the classic antibodies in muscle is myosin, which is that contractile protein. So I went into the refer, and I was going to get some antibodies and stain tissue, and I did all my staining. And we had stimulated muscles that had just been activated. They weren't injured or lengthened or anything. That's what's called an isometric contraction, sort of like just turning it on and turning it off but not injuring it, compared to what I just showed you, which is stretching it while it's activated. And, this isometric guy, you can see, hopefully, I can't really see it too well from here. You can look at my screen later, but all these circles are muscle cells, and the antibody label is turns out for this other protein called desmin, which at the time I didn't even know what it is. It's a structural protein in muscle. And so over here you can see the structural protein is gone in a lot of these cells. So then I had to go in and ask this guy who was in charge of the lab, once I figured out I had the wrong antibody, what's desmin? And he goes, it's sort of, we use it as a positive control for just making sure the structure is intact. This muscle had only been exercised for 5 minutes. So in science and in biology, when something happens quickly, usually you're sort of toward the upstream events of what's going on, initiating events. For immunolabeling to go away in 5 minutes is really interesting because it means you're at the very early stages of sort of what is injury all about. So we've pursued that, and it turns out that Desmond is real important in structural integrity of muscle, but maybe also in signaling what's going on in muscle. And the way you test that these days in biology is to make a mouse that doesn't have any Desmond in it. So we did that, and that turned out to be interesting too. But what I wanted to show you was a cool way to study that whole Desmond process, because I think it's at sort of the junction of engineering and biology. So this is now a picture of a mouse who's hooked up to another little one of our devices that can move ankles. But now he's had his Desmond knocked out genetically, and now what we're going to do is we're going to inject his muscle with DNA that is encoding Desmond. And the idea is you have this Desmond knockout who's got all these deficiencies. If you can put Desmond back in, that's sort of like gene therapy for the mouse. And you can figure out what Desmond does for the muscle. So we're trying to understand its role, but also to be able to use it ultimately therapeutically. So we inject the DNA, we give him a little zap of electrical impulses, and then in a couple of days, the muscle starts making that DNA into protein. So at the top, you can see this GFP is green fluorescent protein, which is a way to visualize where that protein has gone. This is just an overview of the muscle. Here's a slice of the muscle, and you can see some of the cells in the muscle have made GFP and some of them haven't. And so by doing this and combining it with mechanics, we've been able to take, for example, a single cell, which I know is a little hard to see here. You can see this one cell. Remember, muscle cells are big and they're multinucleated, so some of the nuclei are turning out GFP Desmond and some aren't. And the regions of the muscle cell that are making Desmond turn out to get structurally put back into their normal state. And so we can now take these cells, we can do mechanics on them, and we can show, for example, the reordering of the muscle cell that has Desmond put back into it. And I hope you can maybe appreciate where Desmond wasn't expressed, it's very disordered. So we have hope that we can use these therapeutically, maybe for something like contractures, maybe for something like muscular dystrophy. We're really interested in those topics. So I talked to Ebony also about the idea of editorializing a little bit about sort of success in science and engineering. And so I thought I would end with a couple of insights for success, of course, based on my bias. First thing I'd say is clarify your calling. You have to do what you love. We were talking about it a little bit before with the Jacobs scholars. You work so long, 30 or 40 years, you better love it. And you've heard the saying, if you have a job that you love, you never have to work a day in your life. Because if you love it, you'll do it just sort of spontaneously. But you have to clarify what it is you want to do and probably try a few things to see what really floats your boat. For me, it turned out to be science. And I always thought that I would keep this job for a few years and then go get a real job. Because neither of my parents went to college. They both had real jobs. They had to work, and I've sort of never had to have that real job. A lot of scientific success is simply seizing an opportunity when it presents itself. So interacting with hand surgeons, asking their questions, finding people around campus, for example. Everyone here is kind of a genius in their own area. Finding people who do cool stuff and thinking, gee, I wonder if that would work for my system. And developing relationships and just doing it because it's possible. I would say also, I was recruited into an orthopedic surgery department. Not that I was an orthopedic surgeon, but I always kind of liked working on cars and breaking things, which is sort of like orthopedic surgeons think. And so being able to fix stuff was always attractive to me, and hanging around orthopedic surgeons gave me tremendous exposure and sort of an internship in seeing what the problems were and then being able to solve them. So that's another way of saying people and relationships play a huge role in scientific success. You've got to develop your people skills. I really like that the Gordon program is developing leadership skills, because you've got to be technically competent. But most of the world who ends up getting a Ph.D. is going to be technically competent. To have true leadership ability, you've got to be able to communicate, create vision, inspire, etc. And that's one of the things this program is doing. I love this quote from Ronald Reagan: you can get a whole lot done when you don't care who gets the credit. Science is funny that way. We have author orders, and we have PIs, and we have program directors, and that's all important stuff, and people have to take leadership roles. But if you're worrying about who's going to get the credit, it's sort of a deal breaker in a lot of these areas, and I think you have to sort of have a humble, giving attitude, and stuff will just happen. Try not to take your reviews personally. This is sort of a scientific thing. You know, we get rejected. A good scientist should get rejected. If you're shooting high for Nature, Science, PNAS, all those kind of journals, you should get rejected on a regular basis. But if you take it personally, if you get your feelings hurt, and if you go whine and cry in your beer, I mean, it's going to take its toll on you. You got to realize that that's just the nature of the job. If you're not cut out for it, you should get out, because you're just going to get criticized your entire life. Take it as an opportunity. It's a great way. These are all smart people. They have their own perspective. Just let it roll off your back and just go, okay, whatever. I could always get a job at Home Depot or something like that. I like Home Depot. This is a good one for engineers. Don't marry your tool and then walk around and try to see what tools are usable for, what tools can be, what questions you can answer with your tool. It's sort of like if you walk around with a hammer, everything starts looking like a nail. Engineers are famous for developing tools and then walking around and seeing what that tool will do. I mean, ask big questions, develop tools if you need them, use existing ones, but don't marry your tool. That's only going to last you, because honestly, if you have this 30 or 40 year career, your tools are going to be obsolete in a few years anyway. There's going to be new tools. Surround yourself with quality people. I always like to pick students who are smarter than me. Most of mine aren't, I would say. So that's always the good news. Have fun. You should be thankful you don't have a real job. I've always thought that. My wife is in the real world. She's a profit loss person in the real estate world. It's a brutal world. Our job is like a privilege to be able to discover, inspire young people, and hopefully help patients ultimately. So I would say be thankful you don't have a real job. Our work is supported mostly by NIH and the VA. It depends. The VA, you've probably seen the VA hospital over here. They've been very generous. They have a whole rehabilitation engineering division, which generously funds sort of applied engineering to rehabilitation. I'm happy to talk to you more about that. NIH and NIAMS are our major supporters. I also should mention 4 people who have dramatically affected my career. The first one is my advisor, Ron Baskin. I'm talking here now about relationships. Ron was a great thinker and very generous, very open, normal person who basically empowered me to do stuff I wanted to do, even if it wasn't stuff he wanted to do. I've talked to you about Jan Fridén, who's a hand surgeon in Sweden. He and I have been collaborating now for 28 years, and I consider him a brother. We've had kid exchanges, we've had family exchanges, house exchanges, and I've learned a ton from him. Alan Ryan is my running buddy here in San Diego. He's a neuroscientist, very good scientist. He's the guy who slaps me around and says, quit whining, quit all your complaining, you don't have a real job, and tells me how to not take things personally. And you need a buddy like that. You need someone who's going to sort of slap you around and keep you honest. And then a junior colleague, Sam Ward, who just joined us within the past 10 years, but who was a physical therapist Ph.D., who's a great clinician and also a great supporter. So I'd also invite you to visit our website. We have a cool website, muscle.ucsd.edu. And especially with regard to this group, I would say our graduate students here are circled. Trevor is studying muscle scaling. I can't really see the people. Allison is doing some of that ECM I told you about. Is this Lucas back here doing CP muscle? And Gretchen is doing mechanics of that Desmond knockout. Margie's here, but she's not in the picture. I also want to plug our center. We have a National Skeletal Muscle Research Center. We fund pilot projects for people who are interested in clinically relevant muscle problems, or engineers who have cool tools who want to apply them to muscle. So you can check out that website. And my last plug is, obviously you all know the IEM that Dr. Chen is the director of, the Institute for Engineering and Medicine. I agree with the comment that engineering and medicine are natural partners. There's a huge amount of low-hanging fruit that can be picked. And we're excited to have begun the Center for Musculoskeletal Research. Bob Saw and I are directing that. We're trying to get organized. We're crossing departments of bioengineering, mechanical engineering, radiology, orthopedic surgery, and rheumatology, and hopefully medicine. And hopefully you'll be hearing a lot more from us in the future. And for that, I'll say thanks very much.
Thank you very much. So we have time for a few questions from the audience before we then start our panel. And so please, we have a runner with the microphone also.
Oh. Let's see how fast she is. No,
so we have a question right here.
Or comments. I know a lot of you here are leaders in your own field. Feel free to chime in. Yeah.
I was interested in the graph tension and length.
Mm-hmm.
And I'm wondering what's the range in human, the quality factor?
The what? The what factor?
The quality factor. So basically, there is like, it's like a resonance length. It's for example 2 micrometer, right?
Okay, so these are the lengths of the individual sarcomeres, the little machines that power muscle contraction.
Yes.
So in vivo, for example, if I'm flexing and extending my elbow, and I'm wondering how much length change there would be in a biceps muscle, is that what you're asking?
I'm asking what is the maximum tension a human can get.
Oh, okay. So that's a little bit of a tricky question. So I'll give you two answers. First of all, the number we use in engineering for the peak stress of a muscle is 250 kilopascals. But if you measure, if you do almost perfect mechanics and you know the kinematics, say, of the elbow, and you do MR scanning of the biceps and brachialis muscles, so you know their dimensions, and you make really good calculations of the peak force or torque a person should be able to generate, they can typically generate maybe 60 or 70% of that. And that's mostly because you cannot voluntarily activate your entire muscle. So you have built-in circuit breakers that partly are overridden by sensors in the muscle. And part of it is it's a trained thing. So, for example, I don't know if you ever watched the Olympics, those power lifters. I used to think it was such a showboat. These big guys, typically Russian guys, they're walking up to the bar and they're psyching up and they're kissing their hands and getting all ready and powdering up and ready. And then they do this 5 seconds and then they do this explosive thing. Well, those guys are doing all this neural stuff. There's a huge amount of stuff. You can train yourself to change the activation frequency of your pattern, and the higher frequency you turn on a muscle, the more force you get, and you can train yourself to synchronize all the fibers in the muscle. It's tricky. So these guys have a neural training method where they can get like 85, 88% of their maximum muscle force out. And typically that's just a neural limitation. That's why you sometimes hear, the mom lifts the car off her baby's crib or something like that, or some superhuman strength thing. When you get adrenaline, you can change your activation pattern and all that kind of stuff.
Thank you.
Sure. I will tell you though, even though you didn't ask it, it's interesting that muscles do have we do think muscles have a preferred place on this length tension curve that is very muscle specific. Heart muscle is the best example. The heart guys are always ahead of the skeletal muscle guys. I've never really liked them. I think they're kind of, I'm just kidding. Heart muscle, it turns out, the sarcomeres are very stiff and they are relatively short. So as you know, the Starling law is the more a heart fills, the longer its sarcomere gets and the stronger it gets. But there are heart diseases where the sarcomeres are allowed to get shorter, and allowed to get longer, and that's a bad thing because in normal hearts, the more it fills, the more it shortens. The more it fills, the more blood goes out. But if the heart gets messed up, either the connective tissue or the sarcomeres themselves, the more they fill, the less they expel. The more they fill, the less they expel. If they get over on this side of the curve, this is a matter of life and death. So heart has taken great pains to make sure the sarcomeres don't get very long. Other muscles in the bodies are a little bit less stereotypic. Some are very stereotypic, and the one we're studying these days is back muscles. And it turns out that back muscles operate at relatively short lengths too. So as you flex your spine, your muscles get longer and longer, and they get stronger and stronger, and it creates kind of a servo system. And it's actually in balance with your abdominal muscles that are on the other side of this curve. And so we think that obviously it has training implications, but it has significant surgical implications if you're moving muscles around. If you don't put them back the way you found them, or put them back so that they operate on the right part of the tension curve, they just don't work right. And I'm telling you, I'm shocked how well this laser diffraction stuff works, the functional effect it has on patients, and how little it's used in medicine. It's just one of those things. That's the difference, as we were talking about earlier. It's the difference between having an idea that works and a method and having something really be commercially viable.
So for the active curve you have on this chart,
Mm-hmm.
is it real or it should be smooth and
Yes.
passive? Or
Sorry, this is a theoretical curve. So in practice, this is the theoretical relationship that Andrew Huxley and colleagues developed for one sarcomere. In practice, when you do this, since you have hundreds of thousands of sarcomeres in series and parallel, in practice, the typical curve for a muscle pretty much looks just like an inverted parabola. And it all gets smoothed out. Yeah. And so that makes modeling of human muscle a lot more straightforward. Yeah, go ahead.
You just mentioned the fact that the diffraction grading, it can be really useful in the clinical setting, but it's not really been used.
Mm-hmm.
What are some of the challenges that you face, like trying to get it more pervasive?
Great question. I would say we picked hand surgeons because hand surgeons are pretty nerdy as a surgeon. Like the fighter pilot surgeons are the total joint surgeons and the sports medicine people and the people who just go in there, blast through it, and come out of there. Hand surgeons are kind of nerdy and meticulous and whatever. I don't know if you couldn't appreciate it, but when you watch a surgeon dissect muscle, it's shocking how the muscle is destroyed.
So this is another graphic.
I won't show you the graphic one. I'll show you this one again. This guy, this happens to be Jan Frieden, who's doing the dissection. This is just connective tissue being taken off the top, but this blunt dissection of fibers where he actually ends up with intact fibers, and we've done physiology on those fibers, that's very delicate. And it takes an extra, here it takes an extra minute, but when you're doing it for the first couple of times, it probably takes you 5 or 10 minutes. And that's a long time to make a procedure last longer. Then you have to get one of these little laser things, which means that somebody has to make them for you, which is usually me, and I'm not a laser guy. I'm not a laser manufacturer. And we went around and talked to the surgical companies, and they say something like, well, what do you use it for? We go, use it for tendon transfers. And they just look up in the insurance codes and see what tendon transfers pay, and they go, not a big enough market. So there are probably practical limitations in the market of hand surgery, and there are sort of cultural limitations to how hand surgery is done. Those are probably the two biggest ones. I will say the good news is tetraplegia surgery in general isn't very common. There's probably 20 centers in the world. We have trained, we have made it our mission, part of our center's mission, to train the brand new young hand surgeons. So we bring them to La Jolla every other year, and we'll train about 20 of them, send them back to whatever country they go to. Sometimes they go to the US, and we're slowly sort of seeding the field in this area.
Thanks.
Should we do the panel thing?
Yeah.
Okay.
Let's thank the
Okay.
speaker
Yeah,
again.
thanks.